Some of that was in Chapter 2, and I said, don't worry about some of those details, some of those differences between DNA and RNA. I want to go through some of that now, so you do have kind of some notes on that, and you do know specifically what you need to know about DNA and RNA, because all this stuff is going to be important for our discussion. I'm going to do a little lecture on Wednesday of stuff that you can read about in your book, but I'm going to lecture on it instead of just having you read about it, okay? All right, so DNA and RNA. And where do we find the DNA? In the nucleus of the cell. And where do we find the RNA? It too is found in the nucleus, but it's also found in the cytosol. It actually can move back and forth between the nucleus and the cytosol. Basic structure of DNA and RNA. DNA structure would be considered a what? It's a double helix. It's like a spiral staircase. So you got full steps and full railings. RNA, however, it's a single strand. We don't necessarily call it a single helix. It kind of is, but it's just basically a single strand. It's like half of a ladder. One side and half of rungs. What are the basic molecule? What basic macromolecule makes up DNA and RNA? Consists of 3 other molecules that are put together. Nucleotides, which consists of a phosphate group, a sugar, and a base. This base contains nitrogen. In DNA, what kind of sugar is it? What? Deoxyribose. And in RNA, it is actually ribose. Deoxy means it's lost in oxygen. So we have a ribose sugar, and then DNA we have a deoxyribose. We've lost in oxygen. Now, as far as the bases are concerned, what bases are found in DNA? GCAT. So, guanine, cytosine, adenine, thymine. Those are the 4 bases associated with DNA. Does RNA use the same 4 bases? No. RNA uses 3 of those 4. Which 3 does it use? The G, C, and A. The guanine, cytosine, and adenine. It does not use thymine, and in its place we use uracil. Now, I do not care if you know which ones are purines and which ones are pyrimidines. One has 2 rings and one has 1 ring. I don't care if you know that or not. I'm not going to ask you that. I do want you to know what pairs up with what. Now, there's a little law that's associated with DNA that describes how these different bases pair up to form the rungs of the ladder, or the steps in the staircase. What do we call that little law? This is kind of back from Chapter 2 at the very end. Kind of skipped over some of it. The law of complementary base pairing. Which basically says that guanine will always pair with cytosine. Adenine will always pair with thymine. So when we look at DNA, that double-stranded structure, we're going to see that G always pairs with the C, and the A always pairs with the T. Now we know that RNA is a single strand, so we would have a G, a C, an A, a U, an A, a G, a C, a G, what have you. Now I know I said that RNA is only a single strand, but RNA does form temporary pairings with other bases. So if this is the case, would this law of complementary base pairing still apply to RNA? Yeah. And we'll find out on Wednesday how this works. And you can always read ahead, too, in chapter, I think it's, it's learning outcome B, 8, 9, and 10, I think. But in RNA is going to form a temporary pairing with DNA. And the law of complementary base pairing still applies. So G on the RNA is going to pair with the C on the DNA. So here's DNA. Here's RNA. And we're going to have G. Now, we have A here for RNA. What's going to pair up with that A on the RNA to the DNA? T, thymine. But we have a uracil here on RNA. So what's it's going to pair up with on the adenine? Okay? And we'll practice this more on Wednesday. So that's the law of complementary base pairing that says G always pairs with C, A always pairs with T. What is the purpose of the DNA in a cell? Why is it even there? What does it do? What? Okay, it's your genetic code, it's your genetic makeup. And so what is this code used for? How does the cell use that code? What does it do with that information? It makes proteins. It tells when proteins need to be made and how they need to be made. So the function of that DNA is the instructions for protein synthesis. And that's really what drives the behavior of the cell. Maybe it produces an enzyme that facilitates a specific reaction. Or it builds a protein that activates another protein then that changes the behavior of the cell in some way. Okay? So really, it's all about protein synthesis. And we're going to look at how that process works on Wednesday. What else do we need to mention about DNA? What do we call, when you have a section of DNA, there are usually specific, or if you have a whole thing of DNA, usually there's specific sections on that DNA that are instructions for specific proteins or parts of a specific protein. What do we call those segments? Genes. Genes. And genes are specific groups of bases. I mean, they could be tens of thousands of base pairs long that are instructions for specific proteins or parts of proteins that eventually the cell is going to produce. Now, way back when, the theory was that there's 100,000 genes in the human genome. Well, after about 20 years of research, they realized there's only about 30,000. And they're just figuring out how those genes work to make all the different proteins in the body. There's more than 30,000 different proteins in the body. So the question is, if there's 30,000 genes, why is there a million different proteins maybe? So they're trying to determine how some of these genes then can play a role in producing this huge amount of protein. I'm going to show you a video at the beginning of class next week, or on Wednesday, that kind of talks about this a little bit. Okay, it's an older video, but it's still very relevant. So we'll talk about that. Any questions in general about DNA and RNA? All right. Hi.
Hi, Larry's home?
Larry's in the office right by the restroom across the street. If he's not in there, he's in a classroom down the hall, I think. That's all I got for today, unless you have specific questions. All right. Hold on. Right, I always do this. You'll get used to it. I was going to dismiss you, and then I think of something else to say. I did not have my... Long story, I did not have... I'm making excuses, I know. I didn't have a chance to get the full, or to get the next content area, 2.2, online. And I will work on that this afternoon. I know it's due Wednesday. There's not going to be a whole lot of stuff to go over. But what I would like you to do is on your Chapter 3 learning guide, if you have some time today and want to get started on that, I'd like you to at least start doing learning outcomes C15, D16, and D17. And it tells you what pages in the textbook and course pack to read. And that should, if you have, what?
What is that? Where do you get it?
The learning guide. I emailed it to you on Friday, but it's also in the content. It's on Unit 2. It's like the third or fourth thing down. It says learning guide. And you can download that. It's a Word document. C15, D16, D17. That'll at least get you something to do today for a few hours until I have a chance.